Good evening. So I've been asked to introduce a lecture series for this group on agriculture. An offer I couldn't refuse. And I made up this title. I'm going to talk about genetic engineering. I'm going to talk about GMOs. I'm going to talk about that sort of thing. And I'm going to tell you why I'm here. These are nematodes, microscopic worms in the ground. I studied nematodes for 30 years. And 45 years ago, when I was in graduate school, I learned that there's nematodes in the ground and they're really not that great because they attack our crops. And so we have to kill them. We have to sterilize the soil. And that's what I did in graduate school. I was a soil biologist, a nematologist. And then mid-career, I learned molecular biology. I became a genetic engineer, still working with these little guys. And I retired 12 years ago, 13 years ago. And in the last few years, because I'm a scientist, I still read a bit. And I realized there's something wrong with GMOs, with genetic engineering technology. I mean, I read everywhere that it's wonderful, and that's what I said during my career. I was, speaking up for GMOs, for genetic engineering. There is absolutely no way taking a gene from a bacteria and putting it in a plant should result in any kind of toxicity. And yet the rats, lots of experiments with mice and rats show that they're getting sick. So I try to think about what could it be, because really very few scientists speak up, because everybody has a family and a mortgage, you can't speak up. I'm retired, I can't be fired. So 3 years ago, when I started speaking publicly, I put up together a story where basically the only thing that I could imagine was that if you put a bacterial gene in DNA using the technology that we have, and we basically shoot that gene, the gene construct, into the genome, there might be some collateral damage, there might be some proteins that are changed. DNA cannot be toxic, but proteins can be toxic for sure. And I wasn't very happy with my story. And it's only less than 2 years ago that I realized it has nothing to do with the genetic engineering technology. It probably, highly probably has everything to do with a pesticide. So I'm going to tell you a pesticide story wrapped around genetic engineering. It should make for an entertaining evening. 2 years ago, I made a presentation to our city council and didn't quite impress them enough to take me seriously. But I heard the mayor say that there's two sides to this story. And he's correct, there is two sides to this story. There is the side that will explain to you that there is absolutely nothing wrong with genetic engineering technology. There cannot be anything wrong with the genetic engineering technology. And this is what I'm going to show you right now. I'm going to show you a video for a few minutes, 4 minutes I think, of this person who will explain to you what it is.
We are all genetically modified organisms, all 7 billion of us, and every other creature on Earth produced by sexual reproduction. I point this out because genetically modified has become a loaded term that is misused to describe recombinant DNA biotechnology, one of the most important technological advances in the 10,000-year history of agriculture. Throw in bogus, Hollywood-inspired terms like Frankenfoods, killer tomatoes, and Terminator seeds, and you have the makings of one of the most groundless anti-science campaigns in the history of anti-science campaigns. We have been modifying the genetics of plants and animals since agriculture and the domestication of animals began. The anti-GMO activists counter. Nature never moves DNA from one species to another, therefore GMOs are unnatural. Not so. Genes have been moving across species boundaries since life began. Bacteria routinely carry fragments of DNA from one species to another, and that DNA is sometimes incorporated into the genes of the host species. This random movement of genetic material has been one of the driving forces in the development of species. The human genome, for example, has 70% of its genes in common with the sea urchin. Why wouldn't we harness this naturally occurring phenomenon to improve the makeup of our food and fiber crops? Today, 18 million farmers in 28 countries are growing genetically modified crops on 448 million acres, 12% of the world's farmland, or about the same area as all U.S. cropland. It would be at least 3 times that if not for bans and restrictions in many countries. Bans and restrictions that are irrational, anti-science, and terribly harmful to the world's poor. Every credible science, health, and nutrition organization in the world says the genetically modified food available today is safe, without reservation. This includes the World Health Organization, the European Commission, and the Society for Toxicology. How many people know, for example, that the durum wheat used to make most of our pasta had its DNA modified by exposing seeds to high-level gamma radiation, and there is no labeling required? But what about those supposedly evil seed companies like Monsanto? Aren't they trying to control the world's food supply? First, let's remember, these companies are not weapons manufacturers or drug cartels. They're trying to make better seeds for agriculture. Second, the seed companies don't decide which seeds to plant. Farmers make that decision. The fact is, these companies have done massive good. Millions of people have nutritious food to eat thanks to their innovative work that has also reduced the use of pesticides. Recently, the first comprehensive study of genetically modified crop performance reported some surprising numbers. On average, GM crops increased yield by 22%, reduced pesticide use 37%, and increased farmer profits by 68%. In addition, new varieties of GM crops offer nutritional benefits that are not present in the conventional varieties of crops. The best known of these is golden rice, a GM rice variety that, unlike conventional rice, has beta-carotene in the grain. The human body converts beta-carotene into vitamin A. Without enough vitamin A, children go blind and many die, about 2 million every year in poor countries of Africa and Asia. In fact, lack of vitamin A is the biggest killer of children in the world today. Invented 15 years ago, golden rice alone has the potential to end all vitamin A deficiency. Yet the anti-GMO movement is vehemently opposed to it just because it's a GMO. As a result, no country has approved golden rice for commercial farming. So while children continue to go blind and die, the anti-GMO crowd celebrates a victory. And we're supposed to be angry at Monsanto? It's time to put an end to the debate over the safety of genetically modified crops and foods. Today, nearly all of our beer, wine, cheese, and yogurt are made with GM yeast and culture. Most of our livestock are fed genetically modified corn and soybeans, and most of our prepared foods contain GM ingredients. Here's a simple solution. People who want to believe there's something sinister or unhealthy about GM plants should either buy food that is labeled non-GM or pay more for so-called organic food. The rest of us, the ones who embrace science, can get on with healthy and productive lives. I'm Patrick Moore, co-founder of Greenpeace for Prager University.
So I couldn't have said it better myself. I definitely would have arguments with some of what he says. Some of it is exaggerated and some of it is incorrect, but that's okay, beside the point. The point is that probably all of you are interested, intrigued, curious about this issue, this GMO debate. You've read about it. I mean, everybody does. Everybody has read about the debate. There's some people who say, like he did, that it's perfectly fine, and other people think it's like it's frankenfood. It's terrible. It's horrible. So I'm going to give you a little bit more detail. There is a very intense debate, and this is, I just saw this 2 or 3 days ago. This basically perfectly, it's on YouTube. It's the new debate, the 2015 Arthur Rupee debate, where pro-GMO people ask other pro-GMO people polite questions about GMOs and get polite answers, and that's the debate. So I'm going to go a little beyond that, beyond the scene, so to speak. This is the American Academy of Environmental Medicine. Okay, there's a few hundred, possibly a few thousand medical doctors. They're not stupid. They can read the literature. And 6 years ago, as a society, they decided to take a stand and they issued this statement. Based on independent studies, this is 6 years ago already, showing infertility, immune dysregulation, accelerated aging, dysregulation of genes associated with cholesterol synthesis, insulin regulation, cell signaling and protein synthesis, and changes in the liver, the kidney, the spleen, and gastrointestinal tract. And they requested, demanded, suggested a moratorium on engineered food, and as you know, of course, nothing happened. So that's two sides of the debate. On one side, you have some academics and a whole lot of people who will sing the praise of the technology. There's absolutely nothing to fear. And on the other hand, you can find some statement like the one I just showed you. So, I'm going to I don't really have much to say about industrial agriculture per se, other than my whole intervention today. And if you have questions at the end, I'm sure there is time for that. Examples of genetically, this is from the net, somewhere that I found this. Examples of genetically modified food crops include resistance to certain pests, diseases, or environmental conditions, reduction of spoilage, resistance to chemical treatments, like resistance to a herbicide, or improving the nutrient profile of the crop. This is from Wikipedia. Well, I mean, that sounds all very logical and hopeful, and I have absolutely no reservation to say, to approve of this statement. This is exactly what genetically modified food crops include. What this statement doesn't tell you is that over 90% of all engineered plants on the planet today, all engineered crops, all engineered food crops, are of one kind, Roundup Ready. They have been engineered to be resistant to the herbicide Roundup. Over 90%. That doesn't leave a lot of room for everything else. And most of everything else really is on the shelf. It is not commercialized yet. It hasn't seen the day of light. It works in the lab, or perhaps in plots. The other technology is the BT technology, is resistance to certain pests. It's a particular kind of technology which is quite successful, where basically a toxic protein is engineered, bacterial protein is engineered into the plant, corn and cotton mostly. And when the insects, the caterpillars come and feed on the plant, they die. But that is a very small technology compared to the Roundup Ready technology. 10 to 15% of all engineered crops, corn and cotton, are engineered with the BT technology. And of course, they are also engineered to be Roundup Ready, because this is number one, by far. The active ingredient of the herbicide. A pesticide has an active ingredient that does the job, and then it has all kinds of other molecules and chemicals to help it do the job. The active ingredient of Roundup, the herbicide Roundup, is called glyphosate. Glyphosate. You're going to hear that word 100 times tonight. And I made this little pun for GMOs so you can remember it. GMOs are basically almost all glyphosate modified organisms. They have been modified with a bacterial gene that confers a resistance to the herbicide, to the active ingredient glyphosate. So this is it. I'm going to talk about this molecule for the rest of the time that I have here with you. Glyphosate, 3 syllables. Glycine methylphosphonate, glyphosate. It's a very tiny molecule that you can see perhaps in the right-hand corner up there, and you're going to see it on every slide, so you can't escape it. Very small molecule invented in the late 50s, 1950s, by a chemical company called Stauffer Chemicals in the USA. Stauffer Chemicals was in the business of cleaning up industrial pipes and boilers of the mineral deposits that deposit on the sides of the pipes and the boilers. And you can't go in and take them out, you have to use a chemical. So you use a chemical product that will basically grab onto atoms and ions of metals. And if you find a really, really good one, then you patent it. And glyphosate was patented, got a patent in 1960 for Stauffer Chemicals, because it is an incredible descaling agent. That's the jargon word for what it does. It's a descaling agent, a demineralizer. It basically grabs onto metals and puts them back in solution. And it's an incredibly broad spectrum demineralizer. It basically grabs onto all metals in a very powerful way. It's a really, very, really wonderful product. So 1960, well, the word gets around because within a few years, it was known that that molecule kills all bacteria. And it was also known very quickly that that molecule kills all plants. And that was interesting to another chemical company, which was not particularly in the business of agriculture, but they decided to make a herbicide of it. And so they repatented the molecule. They bought the molecule, of course, and they repatented it not as an antibiotic, it kills bacteria, but as a herbicide, because there's a lot more money to be made from a herbicide. So glyphosate was patented in 1969 as a herbicide. And it is an amazing product, as it was an amazing demineralizer. It is the perfect herbicide. It is called a non-selective herbicide. It kills all plants. And it is very powerful too. And it works in many different ways, but the accepted theory of it, the way it works, is that it binds to the metal. This is a protein molecule. Proteins are incredibly complex molecules. Long, long chains of amino acids folded in a particular way to do a particular job. You have 100,000 different kinds of protein doing the job of being a human being in your body. 100,000. Very complex molecule. Most of them are called metalloproteins. We also call them enzymes. And they work because at the center of the molecule, in the center, which I have here in gray and blue, there is an atom of metal, which is why they're called metalloproteins. That atom of metal allows for them to move, to twitch, and do the work. So these metalloproteins have an atom of metal in the center, and I just explained to you that glyphosate was invented to bind to metals. So if you have a molecule of glyphosate next to a molecule of protein, which has an atom of metal in the center, the molecule of glyphosate will stick to the metal. It will steal it, so to speak. And the molecule of protein does not work anymore. So the accepted way that it was found that it kills plants is because there is an enzyme in the chloroplasts, and perhaps some of you know that chloroplasts are really cyanobacteria, so we are completing the loop. In the chloroplast there is an enzyme called EPSPS. It's a part of a not a very big pathway, biochemical pathway called the shikimate pathway, and there is a particular enzyme there that has an atom of manganese in the center of the protein, and glyphosate loves manganese. Suddenly, if you have molecules of this chemical inside the cells of the plant, inside the chloroplast of the plant, that enzyme stops working. And that enzyme is part of the pathway, the shikimate pathway, that makes those 3 amino acids at the bottom of the screen, tryptophan, tyrosine, and phenylalanine, the aromatic amino acid, plus all kinds of other molecules that are absolutely essential to the normal functioning of the plant. And that shuts down. So when you have glyphosate inside the plant shuts down. No more proteins, no more all kinds of other molecules. The plant dies within a few days. It's an incredible chemical, an incredible herbicide. It works extremely well. There are many, many other enzymes that are affected by this molecule as well. And you can't read that, it's much too small, but you'll have to take my word for it, or you can do your own research. Many, many enzymes, like I said, most metalloproteins, all metalloproteins have that atom of metal, and glyphosate loves metals. So it took 5 years for the chemical company Monsanto to do their homework and formulate the chemical glyphosate into a herbicide called Roundup. And when I was a graduate student at the time, Roundup was the new kid on the block. It was the perfect herbicide. It had no toxicity whatsoever. Remember that enzyme in the shikimate pathway that is in the bacteria and in the plants is not in the animals. Animals do not make those aromatic amino acids. Animals don't have the biochemistry to do that. Animals are quite retarded, biochemically speaking. We absolutely do not have the capability of making our own proteins. That's why every day you eat proteins. Otherwise, you won't last very long. So Roundup is invented. It's extremely safe. It absolutely cannot have any effect on animals, including humans. It's a perfect herbicide. It's non-selective. It kills plants, all plants, very powerful, and it degrades very fast in the soil. It's basically biodegradable. It's like you spray the weeds and a few days, a few weeks later, it's gone. Ideal. Well, that was 40 years ago. In the last 40 years, since you're a naturalist club, you'll be interested to know this, since you are interested in the big animals above ground. What I didn't tell you is that the nematodes that I studied are 80% of all animals above ground, below ground. The animals above ground are really negligible in terms of biomass. The animals below ground are much more important. So we have discovered in the last 40 years that of course the bees are affected. Another point, animals are, and perhaps some of you may recognize yourself, animals are basically a long feeding tube, like those nematodes. And some animals have appendages, like legs. You can have 2 legs, or 4, or 6, or 8, depending on what phylum you're in, or wings, or fins, something to move, to go and get your food. But basically, an animal is a long feeding tube full of bacteria that are digesting the food that the animal takes in. When you eat, you are feeding the bacteria in your gut. And then the bacteria feed you. They have a name. We call it the microbiome, and I'm going to come back to that. So the bees have a microbiome, of course, and are disoriented. The earthworms feed mostly on bacteria, and of course they are affected as well. We definitely know that it gives the fish a very severe case of diarrhea. It kills basically all amphibians. We're talking agricultural concentrations here. We know what it does to birds and mammals. Of course, we have a lot of data on mice and rats. So, it kills plants. A herbicide you spray on the weeds because the herbicide kills plants. So you're not going to spray your crops or your garden with a herbicide, you spray your weeds. So if you have a garden or a field and you spray your weeds before you plant, then you can expect some residue of the chemical in the soil and probably also in the crop or in the vegetables that you grow after that. There will be some chemical residue of that glyphosate molecule in the crop. But nothing like this new technology that was invented in 1996. This is a game changer. This is an incredibly successful technology, okay? It has revolutionized agriculture. Roundup Ready technology has revolutionized industrial agriculture. There's absolutely no doubt about it. This is a genetic engineering technology where basically a gene from a bacteria was engineered into the crops using a gene gun, a small machine that looks very much like a hairdryer. You shoot the pieces of DNA that you have worked with, and you shoot them into the plant tissue, hoping that some of them will find their way into the nucleus of a plant and integrate into a chromosome of the plant. Then you disaggregate the tissue, you plate it on some kind of antibiotic, and the survivors will, the survival will be multiplied using all the proper plant hormones into a new crop. You have a successful event. The plant is surviving, you can multiply this plant and make it into millions of acres. Dr. Moore told you that today we have about 500 million acres of genetically engineered crops, 90% of which is engineered to be sprayed with Roundup. So an incredible success. This basically shows you the success of the technology. This is the percentage of corn and soy and cotton. Sugar beet is not on the graph, it would be exactly the same curves. Starting in 1996, this is the beginning of the technology, Roundup Ready technology. Soy and corn the first year, and then cotton and sugar beet within a few years, all the way up to almost 100%. Basically almost 100% of corn and soy and canola and sugar beet is Roundup Ready today, is being sprayed with the herbicide. Roundup Ready means that the plant is resistant to the herbicide, so you don't need to bother about spraying your weeds before you plant. You plant, never mind your weeds, and that gives your weeds more time, a few more weeks to come up. And when your crop is up a foot or high or so, then you spray and you get your weeds that way. And you've sprayed your crop, of course, but that's okay because this is presumed to be completely safe, remember? Non-toxic, period. There is no way it can be toxic to animals. So, Roundup Ready crops have revolutionized industrial agriculture. This is a field of corn, and you can see that it is being sprayed with this molecule glyphosate, with the herbicide Roundup. And the crop will stand, and all the weeds will die. And this is being done for soybean, corn, canola, sugar beet, and cotton. And I think this is what Dr. Moore refers to as Frankenfoods in his speech. So if you spray weeds, you should expect some residue in your crop later on. If you spray your crop, you should expect residues in your crop, of course. But you should expect a lot more, because you are spraying your crop after all. It's not a little bit of residue in the soil that goes into the root system and the plant. You are spraying your crop. Not only that, but because your crop is resistant to the herbicide, it's perfectly okay to spray more than once. You spray as often as you need, within limits, I suppose. So it's perfectly normal for a crop to be sprayed twice, possibly more. So the residues are going up in the crop, okay? And a almost surprising study, fact, showed that when the crops are sprayed with the herbicide, they are resistant to the herbicide. But when they're sprayed with the herbicide, somehow they are depleted of minerals. So this is for soybean, but it also applies for the other crops. So in the grain of soybean, you have less sulfur, or less zinc, or less iron, or what have you. Sometimes a lot less. So your crop is depleted in certain minerals, and you understand why, because they have been sprayed with a chemical that grabs onto minerals and takes them out of solution, so to speak. So this is Roundup Ready, an incredibly successful technology. It's basically, like I said, almost 100% of all those crops are now Roundup Ready, engineered to be sprayed with the herbicide. In 2000 or so, there's a new use for that molecule. A new use, completely new. It has never been done before. You can use the herbicide to spray it on crops that have not been engineered. Now guess what happens when you spray a herbicide like Roundup on a crop that has not been engineered? You kill it, of course. That's what herbicides do, they kill the plants. But if you're a farmer in northern Dakota or Saskatchewan, etc., and the winter is coming, or at least the fall is coming, and it's getting cold, and your wheat crop is not quite mature, you haven't had enough heat units yet, or perhaps your corn is not quite ripe, it's perfectly okay 1 week before to spray your crop with a herbicide to dry it. You're not killing it, you're drying it. You're ripening it. And so it has now become normal and routine and common for farmers to spray a number of crops just before harvest. And so if you look again, if you look at the residues from spraying weeds to the residues spraying the crop to the residues spraying your crop just before harvest, you can imagine that you are getting maximum residues for those crops that are being desiccated. So in Canada and in the US, the cereals are not engineered. They have been engineered, but they're on the shelf. They have not been commercialized, not yet. But for the last 10 years or more, they are now sprayed just before harvest to desiccate, to make it easy. First of all, you kill your weeds. If ever there were some weeds at the end of the season, it's much easier to harvest a crop when the weeds are dry than green, because the combines do not like green weeds. This is a page of the Government of Saskatchewan manual for farmers in Saskatchewan, telling you that you have herbicide options to enhance harvesting. And they will tell you that you can spray glyphosate, Roundup, or a formulation of glyphosate, on wheat, oat, barley, soybean, canola, flax, peas, lentils, dry beans, sunflower, hemp, potato, there's probably a few more. maximum residues of that molecule in those crops when they are sprayed just before harvest. And so the residues have come up from being a herbicide where perhaps you could expect some hundreds of parts per billion in the crop, to spraying the crop where now you're talking parts per million of the molecule in the crop, to spraying the crop as a desiccant, and now the levels are going up. And that's okay, because the EPA and Health Canada 2 years ago raised the legal levels, the acceptable levels of residues in the crops. So from 2 parts per million in a crop, we're now up to 30 parts per million. It's okay. No studies were done. There's absolutely no evidence that it's okay, but the numbers went up by a factor of 10, sometimes 100, for all crops. And so I'm just showing you a few here. Sugar at 10 parts per million, soybean 20 parts per million, cereal is at 30 parts per million. This is the legal levels. The crops are sprayed during the growth of the plants with GMO crops, and they're sprayed at harvest time for all the other crops. But if you look at those 5 crops here, soybean, corn, canola, sugar, and wheat, that is the basis of the diet of many people. You get most of your calories from these crops. Well, here is Dr. Moore again telling you about the safety of the pesticide.
You can drink a whole quart of it and it won't hurt you.
You want to drink some? We have some here.
I'd be happy to, actually, but not really, but
Not really?
I know it wouldn't hurt
I mean,
me.
if you say so, I have some glyphosate.
No, I'm not stupid.
Ah, okay, so you
No, but I know
so it's dangerous,
I
right?
know. No, people try to commit suicide with it and fail
let's-
fairly
let's
regularly.
Tell the truth.
It's not dangerous to humans. No, it's not.
So you're ready to drink one glass of
No,
glyphosate?
I'm not an idiot. Interview me about golden rice. That's what I'm talking about.
we did.
Okay, then it's finished. Then the interview is finished.
That's a good way to solve things.
Yeah. You're a complete jerk.
Okay, so you may think it's ominous because I've been telling you about residues in the crop, and perhaps my tone of voice, yes, thank you. And perhaps my tone of voice lets you know that maybe there's a problem with that, but I really haven't given you any evidence that it is damaging or toxic yet, is it? So I'm coming to that. You remember, glyphosate is the total antibiotic, because bacteria and plants have that same enzyme. They make their proteins exactly in the same manner, and that is blocked, inactivated, and the bacteria die. Glyphosate is a very powerful antibiotic and completely non-selective. If you look at the patent, as I have, there's pages and pages and pages of the list of all the bacteria that are killed. So in 2001, Monsanto, the chemical company Monsanto, filed for a patent for glyphosate as an antibiotic, actually as an anti-malaria agent, because it does kill the malaria agent. I guess they were hoping that it could be used that way and used as a pharmaceutical. The patent was granted in 2010. And in 2013, a study coming out of Germany showed that 1 part per million basically kills all the bacteria from the gut of poultry. Whether it's from the gut of poultry or the gut of any other animal, to me it's equivalent. This is a very simple experiment. Any high school student could do it. So glyphosate kills bacteria. Remember the feeding tube of animals? Full of bacteria that are doing really feeding themselves first and then feeding the animal. So glyphosate, being antibiotic, kills the bacteria, or at least some of them. Damages the constellation of communities that live in that environment. And somehow by killing some or many of those, it doesn't touch some of the pathogenic bacteria like Salmonella and Clostridia. And I often say that, you know, we have more and more cases of Salmonella in poultry houses in the last 10 to 15 years. And we have more and more cases of Clostridium botulinum in cattle, particularly in Europe. And we have more and more and more cases of intestinal infection caused by Clostridium difficile in humans who check out in the hospital, and many of them die because this is very serious. We have no antibiotic to cure Clostridium difficile. So, you as human being are a long feeding tube, and yes, you have appendages to go with that, but basically you're a long feeding tube, and in your feeding tube you have about 100 trillion bacteria, 100 trillion cells. In your belly you have a new organ called a microbiome. It's inside your intestine. When I was a kid, my mother explained to me that I had an intestinal flora and it was important and I had to eat yogurt, etc., etc. It's way beyond that. In the last 5 years or so, the medical establishment has gone crazy over this organ, the microbiome. It turns out that the bacteria in this new organ weigh almost the same weight as your brain. And there are many, many cultures in the world that call this the navel brain. It turns out that the nerve connection from the brain in our head to the brain in our belly, the vagus nerve, is actually the most important nerve connection in our whole body. Those 100 trillion cells talk to each other and talk to each one of your organs. They basically run your body. You don't run your body. You're not thinking about your liver or your intestine or your... It just kind of happens. Who's in charge? Who is doing it? Your organs get all kinds of biochemical messages from your microbiome telling them to do this, to do that, and now it's time to do this, etc., etc. Your immune system, the bacteria of your microbiome are under 100% in charge of your immune system. Asthma, allergies, Crohn, all kinds of immune disorders come from having a damaged microbiome. Back to glyphosate. There's a couple of researchers in the States, Stephanie Seneff and Anthony Samsel, who published an article 2 years ago, 2013, with this title, Glyphosate Pathways to Modern Disease. And they were most interested in celiac. And basically, they say that celiac is probably not caused by, at least the celiac epidemic as we know it today. You all notice that everything has to be gluten-free in the store now. And this happened very quickly, in a matter of a few years. Interesting. Well, they say that probably this epidemic is not so much due as a new sensitivity to gluten, or possibly the crops have been changed, as it is from the glyphosate that is sprayed on the cereal crops. We have known since 1983, this was published in 1983, glyphosate inactivates detoxification enzyme. The CYP, the cytochrome P450 enzymes, mostly in your liver, we have 57 of them in the human body. And they are there to detoxify all the molecules that you ingest, whether there are chemicals in your food, whether there are molecules that are perfectly natural in plants, but somehow we have to detoxify them before we can use them. This is the job of those enzymes, 57 of them, the CYP enzymes in the liver. Glyphosate, and they all have a metal iron in the center. Glyphosate inactivates the CYP enzyme because it binds to the iron. CYP enzymes play crucial roles in biology, one of which is to detoxify xenobiotics. Glyphosate poisoning enhances the damaging effects of other foodborne chemical residues and environmental toxins. Negative impact on the body is insidious and manifests slowly over time as inflammation damages cellular systems throughout the body. This is in another paper published last year by the 2 years ago, sorry, by the same two people, where basically they said, okay, glyphosate as an antibiotic damages the microbiome, and glyphosate inactivates the CYP detoxification enzyme. What could be the consequence of that? And their conclusion, damages the microbiome, inactivates and causes inflammation, and their conclusion is this: glyphosate poisoning causes multiple chronic illnesses and organ malfunctions, including liver and kidney failure, intestinal and immune system failure, multiple cancers, autism, Alzheimer, and dementia. Now, this is not a study. They didn't study it on rats or humans or anything. They just went back to the medical literature, and both of them are biologists and biochemists, extremely knowledgeable. And they say, okay, this is what could be done, and this is what happens if this enzyme doesn't work, etc., etc. This is their paper. They're basically predicting that this will happen to somebody ingesting good quantities of glyphosate. We have this study from 2012. This is out of Egypt, and rats were fed glyphosate in Roundup Ready corn. Rats were fed Roundup Ready corn. Glyphosate in Roundup Ready corn damages the liver and kidney, and this study shows electron microscopy. So electron microscopy pictures of livers and kidneys shows damage. There's another study. I'm showing you a very small sample. There's a lot of studies showing damage. There is also, of course, a lot, perhaps even a lot more studies showing that there is no damage, there is nothing to worry about, and everything is perfectly fine. That's why there is a debate. And this is another study out of France. This team of toxicologists, they work at the Université de Caen, which is where I did my undergrad in France. And I met Dr. Cirellini last year in Regina. They have been looking at Roundup and glyphosate for at least 10 years and publishing paper after paper after paper showing that there is really definitely something very wrong with it. This particular study that I'm showing you there is a 2-year feeding study. That was the first long-time feeding study done by a team of toxicologists on rats. Monsanto, the chemical company Monsanto, had done previously a study for 3 months feeding rats Roundup Ready corn and showing basically no damage. Perhaps a little bit at the end, but that's questionable. These people decided that 3 months was probably not enough, and so they fed the rats for the whole life of the rat, 2 years. They fed them Roundup Ready corn, and they fed them also, some of the treatments had glyphosate in the water to drink. And of course they took all kinds of parameters and blood and urine and measuring everything every week or every so often for 2 years. It's their business, they are toxicologists. And they show that after 4 months, there was definitely some very, very significant damage to the liver and the kidneys. No question asked. And then of course the rats went down, but as time progressed, the rats showed tumors. Now, these toxicologists wanted to basically to replicate the study done by Monsanto. So they used the same strain of rats, and they used the same protocol, the same everything, 10 rats per treatment, etc., as in a normal toxicology study. And instead of 3 months, they did 2 years, and instead of just Roundup Ready corn, they also had the glyphosate and a few other treatments. But after 6 or 8 months, they showed tumors. And the tumors got bigger, and these went on the internet, and this was completely unacceptable to the biotech industry. And so a lot of academics who are basically have grants or somehow money or being influenced by the biotech industry, they have money in the game, wrote letters to the editor saying this is a stupid study, this is not a proper study, the statistics are not done right, they don't know what they're doing, the treatments show that the results are not linear, etc., etc. Everything is wrong with this study. And the editor did something surprising. Instead of being surprised, I would have been surprised at the reaction, say, okay, I'm going to send this paper for another review to another dozen toxicologists. He decided to listen to the biotechnologists who know nothing of toxicology, of course. So Monsanto, the chemical company, made the publishing company an offer they couldn't refuse and put their own editor on the journal, who promptly retracted the paper. So this paper has been retracted because the tumors are inconclusive. Because this is not a carcinogenicity study, this is a toxicology study. So you cannot conclude to tumors. So what do? Are you supposed to ignore them? No, you can't ignore them, obviously. I think Dr. Seralini did the right thing. The paper has been published again, of course, in another journal. And I'm just going to go quickly here because I was only given an hour. 1 part per million reduces testosterone. There is definitely an endocrine disruption effect of glyphosate on human cells. These were rat cells, human cells, endocrine disruption at 0.5 part per million. 0.5 parts per million, you are getting an effect on hormonal balance and equilibrium in human cells. And then finally another study, this is done, this is out of Bangkok in Thailand, Bangkok University. Breast cancer cell proliferation was accelerated at parts per trillion. Now a part per trillion is 1,000,000 times smaller than a part per million. And you get an endocrine disruption effect and a cancer-causing effect at that level. Quickly, there's a long story to this paper. This is from Argentina. Basically, they showed birth defects in frogs from the chemical. Dr. Andres Carrasco, who published the paper in 2010, was the head of the embryology department in Buenos Aires. And in Argentina, until 10 to 15 years ago, they grew grass for pastured cattle. And they sold the meat to the European market and the North American market. But that really does not provide Monsanto with anything, so they changed all that. And so now the pampas, huge plains in Argentina, are not growing grass anymore, they're growing engineered soybean to feed cattle in Europe and in North America. Engineered soybeans, which are, because the acreages are in the thousands of acres, engineered soybeans which are sprayed by plane with the Roundup herbicide, of course, as they should. And so people are getting sprayed in the villages, and babies are born with really, really weird malformation, congenital malformation. And so the word came all the way to Buenos Aires, and Dr. Carrasco went to the Pampas to investigate and saw what was happening and saw what kind of chemicals were being sprayed, came back to his lab, and because he was an embryologist working with frogs, he injected frog eggs, embryos, with picoliters, 10 to the minus 12, picoliters of glyphosate, incredibly tiny, 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 tiny quantities, and was able to replicate in frogs the same symptoms that he saw in human babies, being born with half a head or half a brain, etc., etc. Birth defects in frogs and in humans. Birth defects in pigs, same story, this is out of Germany. The pigs are fed Roundup Ready corn, and the piglets, some of the piglets are born deformed. Another study from Germany where they looked for the movement of glyphosate once you have ingested it, whether in cattle and other animals or humans. And they found that, contrary to what industry says, industry is very clear that if you accidentally ingest some of this molecule, glyphosate, you flush it through. You pee it the next day. This paper, these people are saying exactly the opposite, that glyphosate actually accumulates in basically every organ. The bone, the heart, the liver, intestine, etc. And the same paper had this interesting observation. People with chronic illnesses always had higher residue of glyphosate in their urine. This is a study that was published less than a year ago in December 2014. And the title is Genetically Engineered Crops, Glyphosate and the Deterioration of Health in the United States of America. And what these people did, Nancy Swanson is in Seattle, and what she did is that she basically went to find the statistics of the Center for Disease Control in Atlanta, Georgia. The Center for Disease Control in Atlanta, Georgia keeps tabs and statistics on the health of American citizens. How many people this year in the USA in 2014 checked in the hospital with liver failure? How many kids were diagnosed on the autistic spectrum this year, last year, and for the last 20 years? And all the statistics are there available in the USA. So she took that body of numbers and then she put it next to another set of numbers from the USDA, the US Department of Agriculture. And the US Department of Agriculture, of course, keeps a tab on how many millions of acres of engineered soybean were planted this year, and how many tons of glyphosate were sprayed in the US this year, etc., etc. And she put the two together to do a correlation analysis. You can take two bodies of data like this, and the statistical tools are there to see if the two bodies of data are linked. Is there a cause? Is there a linkage? And of course, what she wants to show is there a link between the rate of glyphosate climbing, the rate of glyphosate applied and used in the USA and spread on the crops, and the rate of particular diseases. And she looked at all kinds of diseases, and I'm going to go through them with you. The first one she looked was autism. So this is the curve for autism. And the red curve is the amount of glyphosate that is being used in the USA. And as you saw earlier, because the crop, the corn and soy and canola and cotton are going up, and now we have the desiccating crops, we're using more and more and more. And there is more and more and more in the food, of course, residues. And so when you do a correlation analysis, you end up with a few numbers. One of them is called the correlation coefficient. It's the R value. And the R value, maximum value, is 0.99 or 0.999, depending if you want it. It's basically the maximum. It's the best fit. And it's pretty obvious here. The yellow bars are the number of children diagnosed on the autism spectrum in the last 20 years, from 1990 to 2010. And you can see that the red curve kind of goes over the top of the yellow bars pretty well. It's a really good fit with an R value of 0.99. And I showed you those two papers from Dr. Seneff and Samso on glyphosate and celiac, and glyphosate and the cytochrome P450, etc. And this is Dr. Seneff saying a few words about this.
Starting out with this, the autism epidemic, back in 2007, I got really worried. I've been watching the autism rate steadily climb over the last 20, 30 years. 2007, I said to myself that I need to take a look at this. Somebody needs to figure out what's happening here. There's clearly something in the environment, probably multiple things in the environment that are causing this. And as a biologist, I maybe could try to figure that out. And so I studied it. I watched the rate go up and up. Now, 2009, 1 in 100. And the most recent number, March 2013, 1 in 50. This is getting to be a very, very worrisome number. And autism is not a trivial thing to have. For those of you who know people who have children with autism, it can be a very severe disability. So I project at this rate 1 in 2 by the quarter-century mark. So half the kids born will be on the autism spectrum if we stay the course. So we need to figure out what it is in the environment that's causing this, and we need to fix it. Very clear to me.
So that was autism, this is celiac. Obviously not trivial to many people either, if you are affected. A lot of people think that they are gluten sensitive, but really it probably is something else. So you can see that the R value is also pretty high. And again, remember, this is about the convenience of spraying your crop just before harvest, so that you have maximum residues of that molecule in the food. It's really not different than, you can spray your crop a week before it's on your plate, or 2 weeks before it's on your plate. It comes to the same thing. You might as well spray your hamburger. It is that. And another video.
A second genome active in and on our bodies. And we've learned that you can sort of classify the gut flora into 3 kinds, 3 corners of ecosystem space. And we've called them enterotypes. And so to give you a better feeling what these enterotypes are, I often make the what's the word? The comparison, thank you, with an ecosystem, right? With a forest. You have tropical forests, you have temperate forests, you have bamboo forests. They're all forests, but you have different species living together and functioning as a unit. You have constellations that work optimally, and that's what I think these constellations in your gut flora are like. And so the environment that these bacteria live in, they determine these constellations, it seems. And the environment in the gut is the food that you eat. And so people have discovered that the people that have more fat in their food, or more protein in their food, or more carbohydrate in their food, they have different gut compositions. And that's important, because more and more diseases are linked to disturbances of your gut flora. Diarrhea, diabetes, obesity, arteriosclerosis, colitis, Crohn's disease, even autism, all have been associated with disturbed gut floras. And it's not merely associations. Bad gut floras can actually cause disease. If you take the flora of an obese mouse and you put it into a germ-free mouse, so one that doesn't have a flora, that germ-free mouse becomes obese. So we're thinking, right? So we can learn something from this flora about your personal health. We're moving towards diagnosing people on the contents of your gut. And so this is being done, for example, for diabetes or for colon cancer. But we can do this in everyday life.
If there's one thing that I want you to remember other than the toxicity of glyphosate, it's of the primal importance of what's in your belly, the microbiome. I learned of that in the last 2 or 3 years, and I'm just blown away. I'm fascinated with this stuff. Okay, we are going down the list. Autism, celiac, and you just heard diabetes, obesity are all linked to the microbiome. Intestinal infection, I talked about Clostridium difficile earlier. And look at the R values. The correlation coefficient are all extremely high. Liver, kidney failure, Alzheimer, dementia, same thing as autism. Definitely the brain is a very sensitive organ. If the microbiome is damaged, the liver, the kidney, obviously the intestine are extremely sensitive organs, but the brain also is on the list. I mean, after all, the neurotransmitters that are in your brain, a lot of it comes from your microbiome. You know that? Do you know that 95% plus of the serotonin in your brain comes from your intestine? If your intestine is not happy, you're depressed. You don't have enough serotonin. You start with depression and then you go on to mental illness, dementia, Alzheimer, etc. I have here, from here to the bottom, a whole series of cancer. There's correlation with all kinds of cancer, kidney cancer, liver cancer, thyroid cancer, etc. That was interesting. In 2015, perhaps some of you heard of it or read about it, the World Health Organization decided that to classify glyphosate as a probable carcinogen. They have enough published evidence. Obviously, they hired a team of experts who combed the literature and decided that there's plenty enough evidence to classify glyphosate as a probable carcinogen. But in all the noise that came with that announcement, I also find that in 1985 the EPA already had classified glyphosate as a possible carcinogen based on studies. But in 1991 the EPA reversed that classification to non-carcinogenic under the authority of Mr. Michael Taylor, who was the vice president of Monsanto a few years later. So I'm going to end here and give the last word to Dr. Seneff.
And I can hope that enough people will start to become informed and people will start to realize that we cannot, this is not sustainable agriculture. The path we are on is destructive. We have to change the way we grow food if we're going to not end up having every well person in the world spending all of their time taking care of the people who are sick and needy. I mean, we have to do something different. So here's what Don Huber has to say.
This is Dr. Huber, Professor Emeritus Donald Huber from Illinois, Purdue University. This is what he wrote. When future historians write about our time, they're not going to write about the tons of chemical that we did or did not apply. When it comes to glyphosate, they're going to write about our willingness to sacrifice our children and jeopardize our existence while threatening or jeopardizing the very basis of our existence, the sustainability of our agriculture. So this is not about our mayor taking one side or another, and this is not about a handful of growers growing GMO corn in the valley. This is about the food that you eat every day, the food that you find in the store. That's the story. Thank you.